Okay guys, welcome back. We're going to keep going with our series on the digestive system, and now we're going to talk about the liver and the gallbladder. So let's start off with some anatomy. Now, first of all, have a look at the liver. The liver is it's quite large actually. It's a very firm, reddish brown organ, and the liver has an incredible amount of metabolic functions. It also has a lot to do with synthetic functions. That means to make things. That we get from our food, for example. We digest it into smaller bits and pieces, and the liver is the organ that helps put some of these things together to give us our more complex molecules. We've got the liver divided up into two principal lobes. We've got a right and a left lobe. The left lobe is quite a bit smaller. The right lobe is further divided into the quadrate lobe, which is inferior below, and the caudate lobe, which is superior or above. That's the division of the liver. We're more or less worried about the physiology as opposed to this, so we're not going to focus on this too much, but let's get further down into it on a different scale. Now, if we have a look at the anatomy and we get a little bit finer, we're going into our microscopic anatomy now. We're going to look at those big chunks of the liver divided up into lobes, and we're going to look at these microscopic lobules. Now, in each one of these liver lobes, we're going to find around 100,000 lobules. And a lobule is going to be the functional unit of the liver, okay? And here's what they look like under a magnifying glass, under a microscope. You're going to have a lot of these cells that are here that are working together to carry out a common function. You'll have a central vein. The liver has a tremendous blood supply, so we'll have lots of venules and lots of arterioles moving through the liver, lots of capillaries, and we'll also have some bigger branches and ducts that move through the liver at all. But so this six-sided figure right here, that's going to be one lobule, and each one of these lobes are going to be made up of hundreds of thousands of these lobules. So these are the functional units of our liver. This is where the main liver functions are going to take place. So we've had a look at the anatomy. Let's go into the physiology now. What does the liver do? What are its functions? And one of the things that the liver is known for is production of bile. And we've already talked about bile a little bit when we talked about absorption of fats, but bile has a big role to play in fat digestion. Metabolic regulation, so how our body deals with nutrients, is all controlled at the liver. The storage of vitamins, the vitamins that we absorb through our diet that we take in are generally stored in the liver. They're produced. Some of these are manufactured a little tiny bit or modified in the liver, and we store them there as we need them. And then finally, the liver is going to have a major role to play in waste removal. Okay, so let's look at bile production. Hepatocytes. Now, let's break that word down. Hepato is going to be something that describes the liver, and cyte, of course, means cell. So hepatocyte are going to be liver cells. So those are going to be the cells that make up those little lobules. Those are the cells that are going to secrete the bile, okay? Now, bile is going to be mostly water, but in that bile, we're going to find bile salts that we've talked about already. We're going to find cholesterol. We're also going to find pigments, and there's going to be some iron mixed in there as well. Now, what bile salts do besides the lipid absorption that we talked about, helping to get those fatty bits and pieces to form into a micelle, is they're going to emulsify lipids. And what that means is they're going to break them down into smaller and smaller droplets. As that happens, we're going to have more surface area to work with, so we're going to have a bigger area for our enzymes to work, which is going to help us increase the rate of digestion. So not only do they emulsify the lipids, but they'll also help when it comes time to absorb them. Okay, so as the hepatocytes go and create the bile, it needs to be collected. And we're going to have some ducts that run through the liver that are going to collect the bile as it's being produced. So we'll have left and right hepatic ducts, and those hepatic ducts are going to collect the bile from, you guessed it, the left and right lobes of the liver. So here we've got our left hepatic duct, and that's going to collect bile from the left lobe, and then over here we've got the right hepatic duct, and that's going to collect the bile that's formed in the right lobe of the liver. Now these two ducts are going to come together, and that's going to form this common hepatic duct. And that common hepatic duct is going to travel between the left and right hepatic ducts down to where we get into the meeting of this cystic duct, which we'll talk about in a second. But the common hepatic duct is the one that's actually going to leave the liver and bring the bile to the small intestine. So here's a closer look at this system of ducts that go through the liver. We get our right hepatic duct here, our left hepatic duct. They come down here and form into the common hepatic duct. At this juncture right here, we've got a branch that goes the cystic duct, which travels to the gallbladder. Okay, now the gallbladder we'll talk about in a couple of slides, but the gallbladder is important in terms of storage. So the gallbladder is right here. It connects to the common hepatic duct through the cystic duct, and then the common bile duct, or the common hepatic duct, sometimes called. This is the one that's going to empty down into the duodenum or duodenum in the small intestine. All right. Another important function of a very, very important function of the liver is metabolic regulation. Okay, there are so many intricate biochemical reactions that happen in the liver. There are so many very complicated metabolic pathways that take place in the liver. It's an absolutely fascinating organ to study from a biochemical point of view. So we're going to talk about some of these things here now. But one of the major things the liver will do is regulate the composition of blood. What's circulating in the blood is generally regulated by the liver. All of the blood that leaves the digestive tract, so everything we eat, everything we absorb across our digestive tract, it gets absorbed into our blood, and that blood travels directly to the liver. Okay, so anything at all that you eat is going to be fixed or filtered or processed or in some way handled by the liver before it enters full circulation in the rest of the body. When the liver is doing this, of course, it's going to be able to extract nutrients or toxins before those things reach the rest of the body and can cause in the case of toxins, for example, before it can cause any major damage. It's generally met at the liver and then dealt with as best as our body can, depending on what the compound is. So these are some of the really super important functions of the liver. Let's give you a couple examples about how our liver helps regulate our metabolism. So let's look at carbohydrate metabolism first, and our best way to look at this, simplest way, is to take glucose as an example. Okay, when your blood glucose is low, for example, when you're fasting, you haven't eaten for a while, you still need glucose to circulate in your blood. Your brain exists only on glucose. It doesn't have any other means for energy. So we need to have a certain amount of glucose in order to fuel the brain. When blood sugar is low, we need to find a way to get blood sugar increased. And the way we do that are the hepatocytes in the liver will break down glycogen. Now, glycogen is a compound that we use in animal bodies to store glucose. So when hepatocytes break down glycogen, what that's going to do is it's going to allow the level of blood glucose to increase. And that way it maintains a constant or pretty controlled volume in the bloodstream. And that makes sure that our brain has enough glucose there to function, has lots of energy. This process, this whole metabolic pathway, is controlled by glucagon, which is a hormone that you guys talked about last semester, produced by the pancreas. So if we look at the figure over here, we're talking about a case of low blood glucose level. When those glucose levels decrease, a message is received in the pancreas that there's low blood levels. It releases a hormone called glucagon. Glucagon will have its receivers in the liver. Once the glucagon arrives in the liver, the message is to break down glycogen, and then when it releases glycogen, when it breaks down glycogen, it releases glucose, and that glucose travels into the bloodstream to bring blood glucose to a normal level. So this is just one example of how the liver controls metabolism. When we look at this from the opposite point of view, when our blood glucose is high, so after we've just had a big meal, our hepatocytes have lots of glucose in the bloodstream. So we're in this case over here now. we need to get rid of that. We don't want to have too much glucose in our blood. So we send a chemical to the pancreas. The pancreas gets a signal. It realizes there's too much blood glucose available. What it'll do then is it'll secrete a hormone called insulin. The insulin will have its receptors in fat cells, and what those cells will do is they'll take glucose from the blood and they'll store that in glycogen that we store in the liver and our muscles, or we'll store it as triglycerides, which we store as fat. And so this metabolic pathway is controlled by insulin. So these are two antagonistic hormones that have an effect on the liver, which helps the liver process carbohydrates. Now, for lipid metabolisms, we're breaking down fats. The hepatocytes will regulate how much triglycerides, how much fatty acids, and how much cholesterol are in the blood. If lipid levels are low, then our liver will release chemicals that will break down stored lipids. So it'll break down fat, and it will release those lipids into the bloodstream. That'll cause an increase in lipids that our cells will need. Remember, lipids are key for making membranes, and that's going to be key for organelles and everything like that that's involved with membranes. So this is a very important metabolic regulation. If lipids get too high, for example, and there's too much of it in the blood, then they'll remove from the blood and they'll be stored as fat until we need them later on. We can also have amino acids that are in our bloodstream, and the liver will control these as well. We don't want to have too many amino acids, so the liver will remove excess amino acids, and then we can use those to make new proteins as we need them. And don't forget, enzymes are proteins, and so we need a ton of enzymes to keep our metabolism functioning healthy. So synthesizing new proteins from amino acids from our diet, that's a big part of what the liver does. Also, if we have too many, then we can convert those amino acids into lipids or glucose, which will be stored in fat, and we can store them for later, perhaps when our diet isn't as good as what it is right now. These are all these really intricate metabolic pathways that are controlled by the hepatocytes in the liver. The liver also has a tremendous capacity to store vitamins. So our fat-soluble vitamins like A, D, E, and K, as well as B12, they're all absorbed from the blood and stored in the liver. So if we are eating quite well and we have quite a healthy diet, and we have an excess or more of these vitamins than we need, then the liver will store them. And then maybe later on our diet's not as good, or we're fasting, or we don't have access to these vitamins readily, then the liver can release those vitamins so that our metabolism stays healthy and functions normally. Another thing the liver is very, very good at is waste removal. One of the things about protein metabolism is that occasionally we will get ammonia formed from breaking down amino acids. Ammonia, of course, is toxic, and what the hepatocytes are very good at is taking that ammonia and converting it to urea, which is not nearly as toxic as ammonia, or much less harmful. The urea will circulate in our blood, and it'll eventually get excreted by the kidneys when we produce urine, and that's what we're going to talk about in our next unit. Another thing that the liver is very, very good at is inactivating drugs or toxins. Many drugs that we take very commonly, for example, a lot of the world drinks alcohol, wine, or beer, or spirits. If we didn't have our liver there to detoxify a drug like alcohol, then every time we drank alcohol, or the time that we drank alcohol, it could be fatal, it could kill us. So inactivating drugs, breaking down toxins before they cause major issues, those are all super, super important things for the liver. It can also alter steroid hormones, which is something you guys probably would have talked about in the endocrine system unit in our last course. Now, the next thing we'll talk about is the blood supply in the liver. The blood supply in the liver is quite unique, and that'll be the subject or the start of our next video.